Good afternoon, everyone. So in my talk, I'm going to talk to you guys about the human cervix and vagina chip models that we have developed in our lab to model the human host microbiome interaction, as well as the physiology of the mucus. So in the female reproductive tract, the two main organs in the lower part of the, So the two main organs of the lower female reproductive tract of the cervix and vagina, and their main role is to provide a gateway to protect the upper female reproductive tract from the direct exposure to the outside environment and the pathogenic invasion. And also to regulating the passage of the sperm during the fertilization time, but more importantly, to protect the upper reproductive tract from the pathogens that I mentioned. And cervix and vagina do this function through these two main approaches here, in addition to anatomically constrain the access of the outside environment. As you see, the channel here is pretty narrow to separate the uterus from the outside, but they do it through these two main approaches that I have mentioned here. The physical barrier that's done through the epithelium, the impermeable epithelium that covers the surface of the vagina and the cervix, and also the mucus layer, which is an important component in the female reproductive tract. The cervix tissue is basically the factory of the mucus production in the female reproductive tract. It provides a physical barrier on the surface of the cervix and vagina that separates the interaction of any particles and substances that want to reach the epithelium, and that protects the epithelium and the upper reproductive tract from those invasions. The mucus layer also consists of a lot of antimicrobial properties that also contributes significantly to the health defense mechanism of the reproductive tract. The other main component here is the host microbial barrier. As we know, on the surface of the epithelium, we have a microbiome, microbiota, a population of the bacteria that are residing on the mucosal layer. And in the female reproductive tract, the most abundant type of bacteria is Lactobacillus species. And these Lactobacillus species, they maintain a stable and healthy symbiotic with the epithelium, which the epithelium provides them with nutrients, and they in exchange provides the antimicrobial properties against other pathogens. For instance, the Lactobacillus crispatus is a lactic acid bacteria, so it produces a lot of lactic acid in that microenvironment, which reduces the pH of that environment, make it acidic, and as such inhibitory to the pathogens. These bacteria also produce a lot of immunomodulatory components that also contributes to the health mechanism of the cervicovaginal microenvironment. So as I mentioned, a healthy balanced microenvironment in the cervicovagina constitutes of the epithelium, covers with a layer of mucosal layer on top, and all these residential commensal bacteria, which is dominated with Lactobacillus species. The Lactobacillus species constitute of 99% of the bacteria that are there. So they outweigh any other opportunistic bacteria that they want to thrive on that microenvironment, and keeping the population under the control. And this is called the symbiotic microenvironment. But what happens is that once that symbiotic microenvironment is disturbed, then these opportunistic bacteria start to take over. They deplete the population of the Lactobacillus bacteria, and they start to replace that microenvironment with a range of different pathogenic bacteria that are mostly anaerobes. And what happens is that anaerobic bacteria produces a lot of sialidases, proteinases, and mucinases enzymes that what they do, they attack this mucosal layer, they destruct that physical barrier, they make direct contact with the epithelium, which colonizes inside the epithelium and then calls a lot of immune cells to come. Infection happens, then these pathogenic bacteria colonize there, form a thick biofilm on top of them, which makes them very difficult to get rid of. This condition, the unbalanced dysbiotic condition, clinically well known as bacterial vaginosis. Unfortunately, BV affects over 25% of women in their reproductive age, which is like just in US, about 21 million women. And unfortunately, the only clinical treatment that's available is antibiotic, metronidazole, which what happened is women get that treatment, They go back, they're good for some time, but 60% of them come back within 6 months. And this condition never gets completely removed. And as Laura mentioned very nicely, it has various health adverse conditions, including preterm birth that Laura mentioned, spontaneous abortions, higher chance of getting HPV, HIV, and other health inflammatory diseases in the pelvic. So the big challenge here is, the problem is we don't know what's going on there. That microenvironment is very complex because there are different factors contributing to the physiology and pathophysiology of that microenvironment. And for years, I would say over 70 years, this condition is known, but the pathophysiology of it is really not understood well. And as such, they can't come up with the better treatments. So what we did at WIS, we decided to make organ-on-chip, use organ-on-chip technology to make human vagina and cervix tissue organ-on-chip models to look at the host microbiome interactions. And we used the emulate dual-channel microfluidic device and recreated the epithelial stroma interface on this organ-chip model here. We plated the epithelial cells, vaginals and cervical cells on top of the ACM-coated membrane, and the stromal fibroblast cells on the bottom of the channel. And we used an optimized method that we developed in-house, and within 12 to 15 days, we are able to create a thick epithelial, vaginal, and cervical layer on the chip with supported stromal cells on the opposite side of the channel. We characterize the vagina chip, and the vagina chip shows characteristics of a stratified in vivo-like phenotype. As you see here in the cross-section of the chip, we see a stratified epithelium that has the basal layer and also showed a well-maintained barrier functionality throughout the 15 days of the culture, as we show here for 2 different donors that we tried. The epithelium also shows main markers of the vaginal epithelium, including CK5, CK14, and involucrine. One of the main proteins that the vaginal epithelium secretes is the glycogen. Glycogen is the main source of food for the commensal bacteria in that microenvironment. And we found that the vagina chip produces glycogen that we can collect in the effluent of the chip. And as you see here, increases in concentration with time of the culture. As I mentioned, the cervix tissue in the body is the factory of the mucus, and that's one of the main functions of the mucus in that microenvironment. Sorry, the main functions of the vagina chip, cervix in that microenvironment. So here you see the cross-section of the cervix chip, the side view of the main channel at day 5 of the culture, when we have cells on their expansion. So we have only one layer of the cells there. And as you see, there's no mucus layer formed there. But by day 12 of culture, when we are well in differentiation, we see formation of a thick mucosal layer on top of the epithelium, which is also positively stained with germagglutinin, which is a lectin binding marker that binds to the mucin type 5, the primary source of mucin in the cervical mucus. In collaboration with Dr. Lebrila's lab at the University of California, Davis, we performed some glycomic analysis of the mucus. And here you see a comparison of the N-glycan and O-glycan analysis on the mucus that we collect from the cervix chips as compared to the clinical mucus. And you see here that we can very nicely recapitulate the diversity of the glycosylation of the mucin that we see on the clinical mucus. In collaboration with Dr. Rebeck's lab at MIT, we also developed a method for non-invasively look at the biophysical property of the mucus on the chip. As we know, biophysical property of the mucus is one of the mechanisms of defense in that microenvironment. So our method allows us that during the culture, we can look and see how the biophysical property of the mucus is changing. And in this case, we just looked at the mucus at the end of the culture time, which is like 12 days of culture, and compared that to the clinical human mucus. And we found the diffusion constant of the mucus that we get on the cervix chip is actually comparable to that of the clinical mucus and much better than the mucus that we get from the static transfer culture that is commonly used in studies. Menstrual cycle, we all know what happens. There's changes in the sex hormone levels, including the estrogen and progesterone. And these changes in the hormone levels have a major role in changing some of the physiological functions that are happening in that microenvironment. And they are directly correlated with the composition of the microbiome in that microenvironment, and as such, to the health defense mechanism of that microenvironment. So it is important for us to be able to recapitulate that on the organ chip model. So we treated the chips with two different hormone levels, ovulatory and non-ovulatory. So ovulatory is obviously when we have very high levels of estrogen, and non-ovulatory luteal phase, high level of progesterone. And we observed that in this side view imaging, you are seeing that the thickness of the mucus is significantly higher than that of the one we get in a non-ovulatory phases. As you see here, we're quantified based on the images. Also, biophysical analysis of the mucus showed that when we have non-ovulatory hormonal regime, which is high progesterone, the diffusion property of the mucus significantly reduces compared to the ovulatory phase, which is similar to the trend that we see in the human clinical mucus as well. And this is pretty obvious because during the ovulatory phase, the transfer of the sperm, the environment needs to be ready to transfer the sperm, so it becomes more diffusive. In addition to that, we looked at the glycomic analysis of the mucus. And this is, again, in agreement with what is observed in vivo, that in general, when we are in the ovulatory phase with high estrogen, we have higher abundance of O-glycosylation and N-glycosylation on the mucus compared to the non-ovulatory phase. We also have developed an organ-on-chip system in-house that we integrate transepithelial electrical resistance sensors on them. So we can dynamically monitor how the electrical resistance of the epithelial layer is changing with time. And we use that for our system here when we are developing the cervix chip model under the two hormonal conditions and look at how the barrier function is changing. And as you see here by day 11 of culture, we see a significant difference in the barrier of these two models. And that is again in agreement with what we observed in vivo. During the ovulatory phase, there's higher progesterone. The tight junction loosen up because they want to allow transfer of more fluids from the interstitial tissues to the cervical lumen to increase the water content on that microenvironment. So after we characterized the two models, the next step, of course, we wanted to look at the host microbiome interaction. That was the main goal in the beginning. So in collaboration with Dr. Ravel's lab from University of Maryland, we used clinically isolated optimal Lactobacillus crispatus strains that were collected from women that have a stable Lactobacillus dominant communities for years. And they were collected and we inoculated the vagina chip with either a single strain Lactobacillus crispatus or 3 different consortia of 3 or 4 of these strains. So we inoculated the vagina chip with these different bacteria and co-cultured them for up to 3 days. And we observed that at the end of the 72 hours, we are able to maintain a stable population of the Lactobacillus crispatus consortia on the chip compared to the inoculum. Interestingly, when we have Lactobacillus crispatus population on the vagina chip, the pH level of that microenvironment is maintained within 4, which is an acidic level, and it is comparable to the pH level in the vagina in vivo. Also, the strain ratio analysis showed that the 3 days of co-culture of this consortia on the vagina chip does not change as much the ratio of these different strains that we are using on the chip. In fact, the microenvironment that we have created on the vagina enables for maintaining of this ratio between different strains. Also, cytokine analysis shows that the presence of the Lactobacillus crispatus and its colonization for 3 days significantly reduces the pro-inflammatory cytokines here, presented here as IL-6, IL-8, IL-1 alpha, and IL-1 beta, both for the consortia as well as for the single strain. Also, we know that the lactic acid, as I mentioned, is one of the main components that the Lactobacillus crispatus secretes in that microenvironment and contributes to the defense mechanism of that, the cervix and vagina environment. And we found that we can tell the difference, So when we have among these 3 different consortia that we tested, 2 of them were found to actually induce production of D-lactate. The lead lactate is one type of the lactic acid that's specifically contributing to the defense mechanism, and we could recapitulate that on the chip. So we established a model for a symbiotic microenvironment, and we wanted to see if we can model a dysbiotic microenvironment as well. So we used this isolated dysbiotic bacteria, again, from the women that have dysbiosis, and we come up with 2 different consortia called here BVC1 and BVC2. The BVC1 is a consortia that consists of 2 strains of Gardnerella vaginalis and Prevotella bevia and Autopobium vaginae, and the BVC2 is just the 2 strains of Gardnerella vaginalis. And again, we co-cultured them with the vagina chip for about 72 hours, and we found that we can maintain the population, this population of bacteria on the chip for both of these consortia for the 72 hours. And the pH level on the vagina chip for one of these consortia significantly increased. This is interesting because in the clinic, one of the major diagnostics approach is to just measuring the pH. And once the pH on the chip in the patient is high, that's one of the indications that they have developed BV, and we were able to recapitulate that on the chip as well. And as you see here, the presence of these dysbiotic bacteria also caused cytotoxicity. The number of the mammalian cells also significantly reduced. And also cytokine analysis shows that with the presence of this dysbiotic bacteria, we see a surge of inflammatory response from the epithelium for all of these 4 cytokines that we looked at. So we also went ahead and developed the symbiotic and dysbiotic model on the cervix chip. And here you see that we selected one of the L. crispatus consortia and one of the dysbiotic consortia, mainly the G. vaginalis, inoculated them on the cervix chip and maintained for 3 days. And here you see that these models, we are able to, on a daily basis, look and see how much bacteria are actually coming out of the chip. So this enables us dynamically monitor what's going on the chip. Here you see the quantification of the bacterial load in the effluents as well as at the end on the chip. The pH level, interestingly, on the cervix chip didn't change as much, either for the C6, which is the Lactobacillus consortia, as well as the G. vaginalis. And this is, again, in agreement with what we see in vivo. In vivo, the cervix has a lot of mucus, and mucus is known to have a buffering capacity to control the changes, the significant changes in the pH. And we also were able to recapitulate that on the chip. And on the right here, you see a representative bright field image of the cervix chip. It's hard to see here, but I hope you can trust me that there are Lactobacillus crispatus colonized on the epithelium. Here is just the immunofluorescent staining of the epithelium that you see with the presence of the G. vaginalis. The integrity of the epithelium is completely destructed. And the lower number of the mammalian cell is also observed on the chip. And continuously monitoring the barrier function on the chip shows that with the presence of the G. vaginalis, at 72 hours, we see a significant compromisation of the epithelial barrier on the cervix chip. The cytokine analysis also showed that the presence of L. crispatus consortia on the cervix chip minimally changes the cytokine levels on the cervix chip, some improvement with TNF-alpha. But with the presence of the G. vaginalis, we see again a huge increase in all the 4 studied cytokine levels on the cervix. Having the mucosal layer gives us the advantage to look at the interaction of the bacteria and the cervical mucus on the chip. Here you see a cross-sectional image of the cervical layer, and the middle and bottom. The middle one is where the chip is inoculated with the L. crispatus, and the bottom is G. vaginalis. And I hope you can appreciate some of the qualitative changes that are happening in the cervical mucus with the presence of these 2 types of commensal and dysbiotic bacteria. And when we quantified that, we found that the thickness of the mucosal layer, when we have L. crispatus, is significantly higher than the 2 other groups. And also the intensity of the mucus signal is significantly higher than when we have the G. vaginalis. And the biophysical analysis also showed that the presence of the L. crispatus significantly reduces the diffusion constant and diffusion coefficient, diffusion constant and diffusion exponent of the mucus, which both of them indicating that when we have L. crispatus, the mucosal microenvironment becomes more constrained and more confined, which is one of the major defensive mechanism of the mucus. It becomes less diffusive, so the bacterial and pathogens and virals can have minimal activity inside of the mucus. And also the O-glycomic analysis showed that the presence of the G. vaginalis significantly reduces the O-glycosylation profile, and specifically the sialic acid O-glycosylation. And again, this goes back to having G. vaginalis has a solidases enzyme, and that directly cuts off a lot of sialic acid from the mucus. And in summary, I can say that the vagina and chip models that we have made represent physiologically relevant and functional models of the human vagina and cervix tissue in vitro. We can use these models for studying the physiology and pathophysiology of the cervical mucus and the host microbiome interactions, both for healthy and disease situations. And these models also enable us to look into the contributions of the different ethnic backgrounds and different genetic contributions that peoples from different parts of the world have in the disparity of the disease and health condition, as we all know. And also these models are currently being used as potential preclinical models for evaluating new biotherapeutics. And at the end, I want to acknowledge all of my team members at the VIS and also collaborators at MIT, University of Maryland, University of California, Davis, Boston Children's Hospital, and Massachusetts General Hospital, and the Bill and Melinda Gates Foundation for the funding. Thank you very much.